All right. So wanted to try to finish up the PowerPoint on antigen presenting cells um and the MHC molecule and loading and activation of cells and why we have it. So we left off class on Thursday discussing how MHC class 1 molecules are presented and loaded onto every cell type that there is. And again, MHC class 1 are the MHC molecules that activate or are important for activation and selection of CD8 positive cytotoxic T cells. And these cells are the ones that can kill human cells that are aberrant either by viral infection or tumor uh, or have formed tumor cells. And the concept is that you can get cross presentation on MHC class 1 in the event that exogenous antigens come from cells that are human in origin. So when cells die either by apoptosis or necrosis, when a macrophage or a dendritic cell consumes these dead cell particles, they can break down the various antigens from within that and uh, retrospectively or retrograde the traffic the antigens to the rough ER, or they can be cleaved through the immunoproteasome and then loaded onto MHC class 1. And again, when we have MHC class 1, it depends on what the cell type has, because again, if you have a T cell that is looking for this particular receptor and does not match up, because again, it lacks the receptor to recognize this particular MHC and this particular antigen, then that is just uh, a normal function of the immune system to decipher between what is self and what is not self. So as I mentioned before, MHC class 1 and MHC class 2, they have these pocket regions. These pocket regions in between the alpha 1 and alpha 2 subunit. There's a third alpha subunit, and this is the beta 2M subunit. And in between, or in the cleft of alpha 2 and alpha 1, here in this spanning region, again, here is alpha 1, alpha 2, and there's an abridgment between these two alpha helices of anti-parallel beta-plated sheets, and these anti-parallel beta-plated sheets in the inner effacement of alpha 1 of the alpha 1 protein have these varying spots that are hypervariable, meaning that they have slight mutagenesis or changeover of nucleic acids that then translates to different different amino acids in each of these different sites. This variability of amino acids gives the variable region an opportunity to bind to a variety of different amino acids that are presented on the peptides uh, in the epitope form. Because, when you have all these varying antigens on these various different pathogens, there are a great number of different amino acid combinations that could be loaded onto this as well. Now, the interesting thing about this molecule is when a T cell looks to recognize these particular molecules, the T cell actually needs to be able to recognize the MHC molecule itself and the epitope. And the ability for the MHC T cell interface as such, even the absence of epitope, is really important for identification of self antigens, or actually, I should say, recognition whether or not antigens that are, or sorry, epitopes that are loaded are actually epitopes that are not human. And to the point where the makeup of all of these constant aspects of the MHC molecule make up something called the human leukocyte antigen locus. And this defines something called your allotype. And what your allotype is points of contact between your T cell receptor and the MHC that determine whether or not the the MHC molecule that you have actually is yours. And this is the defining feature of host versus graft disease. So this is why you need to have the same blood type and similar MHC molecules or genetic background as someone who wants to donate a kidney or a liver or other type of organ, because your T cells will then look to see if the tissues that have been integrated into the new host body have the same MHC molecules, because again, there is a component that looks at T cell compatibility with engagement with MHC, that's in the constant regions. And then comes the specificity of the T cell receptor variable region in its ability to recognize the variable region of the MHC molecule, which would house the epitope. So again, we have these sites of constant region. That's step one, and all T cell receptors should be able to recognize this. And then finally, we have the epitope, which would then be needing to be recognized by the TCR's variable region. So again, all nucleated cells, including those that are involved in activation of T cells, such as dendritic cells and macrophages that you see here, produce MHC class 1. But how macrophages and DCs and B cells actually produce class 1 is that they also produce a second class of MHC, and this is MHC class 2. MHC class 2 is really important for the ability to, again, activate T cells. In this case, MHC class 2 actually activates a different class of T cells. These are CD4 positive T helper cells. And these cells do two different things. The first thing they do is they're really essential in activating B cells and T cells that are cytotoxic. So the activation of cytotoxic T cells and B cells is a really important part of the function of activation of CD4 positive T cells. The other is the enhancement of the innate immune system. So these also synergize with T helper cells, the enhancement of the function of the innate immune system. Innate immune system. And so because these are, again, MHC class 2 is produced to look at extracellular recognized microorganisms, including viruses, parasites, bacteria, and fungi, because even viruses, they can be seen from within inside the cell, but they can also be seen outside of the cell because they have to dock to the cell to get into the cell. So because each one of these different pathogen types has to have a part of their biology as an extracellular component, they're all possibly exposed to the function of the innate immune cell system, as well as could be targeted at the very least by B cells that would then produce antibodies that will stick to these pathogens. And as we've seen before, the activation of the adaptive immune system and the production of antibodies is really essential for another part of the innate immune system, and that would be the activation of the classical pathway of complement. So dendritic cells themselves, they are stimulated by engagement. They are stimulated either through the binding of certain receptors on their cell surface to molecules which are pathogen associated. And again, these are the PAMPs that we've talked about. And so here's PAMPs. They also can be triggered or activated by pro-inflammatory signals, such as interferon gamma. So much like macrophages, they can be activated by chemical expression as well as direct engagement. This then, if it's going to express or have a stimulatory effect on this dendritic cell, dendritic cells will secrete a small contingent of cytokines, including IL-12, interferon gamma, TNF alpha, and IL-6. And they do so for a lot of reasons. Now, we'll do a whole other lecture on cytokines and cytokine engagement and what these are. But by and large, cytokines come in two families. There are cytokines, and these are involved in immune activation. And then there are chemokines, and chemokines are involved in chemotaxis. And these molecules have certain names, such as IL or IFN or TNF, and they either describe the molecule as a class of molecules. So, for example, IL stands for interleukin. Interleukin, because these, when they were first discovered, were thought to be really important for communication between immune cells. So interleukin, interleukocyte in between immune cells. Interferon is important for interference of the antiviral cascade, at least when we first found it, but then we found other molecules that have the similar structure, so then they became also interferons. And then something like TNF alpha, like tumor necrosis factor alpha, gets its name because when you engage tumor cells with TNF alpha, it kills those tumor cells through the fast cell trail receptor network. So dendritic cells, they play an important role in determining what the infectious agent is or what the indoor stimulus is, secreting some cytokines on their way to activating the T cells of interest, whether they be T helper cells or they could also they also are essential for activating cytotoxic T cells, and they play an important role in skewing the immune system to the specific response that the immune system is supposed to have, also known as the T helper response, and the cytotoxic T cells if they are required. And in the same breath, they also secrete cytokines that inhibit the activation of the anti-inflammatory conditions, such as Tregs. Now, dendritic cells do not secrete the same profile or abundance of cytokines, as we'll see later on in the semester, as T helper cells or macrophages, but they do secrete some of them in order to amplify that signal to activate a very specific subset of T cells that are required to deal with a very specific kind of infection. So, dendritic cells, they play a really important role in consumption of microorganisms, for presentation on MHC, cross presentation on MHC class 1 and 2, but they also are really important, and probably their most important function is that of the ability to activate T cells. So we have different kind of dendritic cells that exist, and they do different things. So, we have myeloid dendritic cells. These are the most common. These are conventional dendritic cells, so to speak. And then we have things like monocyte-derived dendritic cells and plasmacytoid dendritic cells. Um, they play kind of different roles in very specific things. So monocyte-derived dendritic cells are very pro-inflammatory. They typically activate as a function of the secondary immune response. So they kind of activate in response to the activation of the adaptive immune system. Plasmacytoid DCs are very specifically activated in the case of viral or tumorigenic aspects because they secrete type 1 interferons. But again, the one and for the remainder of this semester, when we talk about dendritic cells, we're really talking about these guys, the myeloid dendritic cells. So the myeloid dendritic cells, they play a really important role in again cross presentation, activation of CD4 and CD8 positive T cells, because like again, these are the cells that are going to activate T cells. And if you go back to the first lecture of this class, we talked about how dendritic cells, they load up their MHC molecules with epitopes, and then they travel via the afferent lymph to the common lymph node, where they then travel specifically to the T cell zone, where they then select for T cells that can recognize those particular epitopes and MHC molecules. Now, macrophages, on the other hand, do not migrate. They will drain eventually when the infection is over, but they do not migrate in the first part of an infection. And they don't migrate because they have very good you know very similar to the functions that you see in monocyte derived dendritic cells, the ability to produce things like nitric oxide, high levels of TNF, um, other superoxide production in a event called superoxide burst or respiratory burst, where macrophages can actually produce things like hydrogen peroxide, high levels of uh, intracellular nitric oxide, and these all come together in basically disrupting DNA and oxidizing lipids, and which all make them very good at killing microorganisms or destabilizing viral capsids, or disturbing viral genetic material. As long as the virus, so viruses are a little bit weird because as long as the virus isn't infecting the macrophage, rather the macrophage is phagocytizing the virus, that is where that becomes really important. So as the cells are producing interferon gamma and they're releasing things like DAMPs, these synergize together to form the M1 macrophage, which is really important for killing microorganisms by phagocytosis and increase in respiratory burst, and generating localized inflammation. But the biggest thing that they do beyond being kind of the first line of immunological defense at the cellular level is the cytokine release. And these produce far more cytokines in a much more potent way than dendritic cells do, because these cytokines that they release are not specifically designed for release into the lymphatic system, which is going to help with the activation of those T cells. This cytokine release travels down the lymph to help synergize the release of uh, cytokines and other chemical messengers from dendritic cells, but also they release it into the blood, because this cytokine release is going to dictate what kind of cells in the innate system are going to first arrive to help deal with the inducible innate immune system. So they can induce the expression of more macrophages because that's going to be required. But then, if you have bacteria, fungi, uh, parasites, you need to activate your neutrophils. If you are dealing with viruses, this is natural killer cells. And if you're dealing with things like tapeworms, or multicellular parasites, these are your mast and eosinophils. So macrophages really are found throughout the entire body, and there are very different kinds of macrophages, and they are specialized in different kinds of spaces. So we spend a lot of time talking about microglia, which are the um, macrophages that uh, exist in the brain, and they kind of play this big role in regulating a lot of immunological cascades in the brain, as well as so they do both the preventing infection and dealing with infection and recruiting macrophages if they're overwhelmed to deal with infection, but they all sorry, recruiting immune cells to deal with infection if they're overwhelmed by infection. But they also play an important role in regulating architecture of the neural space, regulating um, the myelination of different neurons, the ability for the neurons to have certain uh, density of dendrites and axons and all these different components. Um, in the liver, we have different kinds of um, macrophages. These are called Kupffer cells. So Kupffer cells are macrophages that are specific to the liver. There are alveolar macrophages. These are macrophages in the lungs. Um, and osteoclasts are a kind of macrophage. They're important for resorption of bone, not necessarily much in the way of infections, but they can play a slight role in those particular infections. Um, and then, in other places, so in the epidermis, this is in the upper skin, we have Langerhans cells. So Langerhans cells have, they are more specialized to deal with skin infections and skin dysbiosis. And then we have interstitial macrophages. These are macrophages you find in the bloodstream. Um, and then, we have other types of macrophages from other places. And the other really important thing to note is that macrophages and dendritic cells both derive from the same general progenitor cell, and that is the monocyte. And so the monocytes that display certain particular receptor markers are ones that are then destined to either become things like macrophages or dendritic cells. So the environment in which they live, the markers that they already possess, and the functions of these things are very much the same. They both play a really important role in regulating inflammation and in determining what kind of response they have, but they do so in a very different way. Macrophages are more of a totality of release of cytokines and activation of both innate and adaptive immune cells, whereas dendritic cells are really just specialized in their ability to activate T cells because for the adaptive system, everything follows that event. Now, macrophages, DCs, um, so macrophages, um, dendritic cells, B cells, these are all um, professional antigen presenting cells which can express MHC class 2, but they're not the only phagocytes in the immune system. Neutrophils also fit this bill. So neutrophils also are phagocytes. So all four of these different kinds of cells can do something called receptor-mediated endocytosis. And this receptor-mediated endocytosis is very much specific for the ability for phagocytosis. So the ability to sense and endocytose whole microorganisms, and in doing so, always having the fate of bringing these particular endosomes to the lysosome or other killing compartments within the cell. So macrophages, other phagocytes, they recognize microbial surface components called PAMPs. Again, these are direct engagements, right? So PAMPs are recognized by certain receptors. So they serve as ligands to these receptors on the surface of these phagocytes. When these receptors are engaged, and this is why complement, if you think about it, this is why complement is also so important. So complement receptors play an important role in this process, because at some level there is also a kind of a minimalist approach, or a minimalist number of receptors that these cells can have. But the point is that these particular pattern recognition receptors, or PRRs, recognize certain molecular patterns called PAMPs, and they allow for like this very specific engagement results in an endocytotic event that always results in the formation of the phagolysosome. But the reason that complement is so important is because complement can actually reach a great other number of membranes, including viral capsids, protists, and fungi. So you get kind of the expansion of potential pathogens that are covered by complement to make up for the sparseness of other pattern recognition receptors. Now, the important thing about pattern recognition receptors is that pattern recognition receptors very specifically activate phagocytosis. And again, phagocytosis is kind of a unique thing in that when there's a direct engagement with a receptor and a ligand, this induces the creation of something called a pseudopodium. So the actual actin cytoskeleton at the membrane pushes the membrane uh, up and around this particular particle, whatever this antigen is, and then completely surrounds it with membrane. So this part here is actually coming from the membrane itself. So this is a phospholipid bilayer, which is why it can fuse to another phospholipid bilayer in the form of the lysosome. There are other mechanisms by which different cells can bring in different things. One such way is micropenocytosis, which is cell drinking. This is a non-receptor mediated actin engagement to the membrane that pushes the membrane around to allow for basically swallowing of extracellular fluid and any particles that happen to be out in space. Now, these vesicles typically are sent to the lysosome, but then we have other types of uh, cell engagement and receptor-mediated endocytosis, or not, such as calveolar and clathrin-dependent mediated endocytosis. And these particular endosomes do not always wind up in the lysosome, because sometimes these particular things will have things you don't want to expose to the lysosome, such as bringing in glucose or other small molecules that are not complex that need to be broken down by the processes of the lysosome. So this kind of function, and why this is important, is because the ability to get this to get the these extracellular phogens and have their epitopes loaded onto MHC class 2 depends on the degradation of that microorganism and its proteins and such in the absence of the proteasome. So the proteasome is good at clipping up these proteins and loading them onto MHC class 1, then that makes sense because it's clipping them to a certain length. And even if you get too high, you have endoplasmic reticulum aminopeptidase that can clip down the peptides that are too long. When we have engagement of the pattern recognition receptors from an extracellular pathogen, this then allows for receptor-mediated endocytosis. And over time, the endosome actually sequesters proteases or has proteases in them as a part of being up close to the cell membrane like this. And as they are brought into the endosome, the acidification of this over time does allow for the activation of these particular proteases. Because a lot of proteases are actually pH dependent in their activation. So the acidification of the vesicle allows for these proteases to then cleave up nascent peptides or even fully formed proteins into nascent peptides and then nascent peptides into epitopes. And then the epitopes are simply loaded on to MHC class 2, which binds to the vesicle via a vesicle-vesicle fusion. So how exactly does this work? Well, MHC class 2 is actually made, again, this is made because you had an engagement in a receptor. So here's my cell. It has a receptor. It engaged with a a PAMP, and then this event then triggered the expression of MHC class 2. And so when MHC class 2 is created because of this engagement, it is made with 2 alpha and 2 beta subunits, and then it is bound to and kind of stabilized by this protein called the invariant chain. The invariant chain is then broken up by another protein called CLIP, and CLIP actually is cleaved to the point where it leaves only a small fragment called CLIP left inside the MHC class 2 molecule. So when the MHC class 2 molecule is kind of circulating around, it's looking for these specific phagosomes that have been acidified. So there's a pH and a trafficking component to this where they find each other, and the CLIP protein is actually removed by the binding of another particular molecule called HLA-DM. So HLA-DM finds these larger vesicles. It infuses to the vesicle in a similar manner that this happens. So it comes in on a vesicle, it's put into the vesicle of MHC class 2. This then opens the 2 alpha subunits in a manner that can then allow for whichever peptide is the right length and the right amino acid context to bind into the variable region, resulting in the recollapse of these 2 alpha subunits around the peptide. Again, much like putting a hot dog in a hot dog bun. And now you have fully assembled MHC class 2 molecule with the epitope of interest. So MHC class 2 has a much smaller kind of placement of variability and its ability to bind different amino acids, but has a very similar structure than MHC class 1 in that we have in that variable region 2 alpha helices that sandwich between anti-parallel beta pleated sheets, but now it's on the beta subunit. So the beta subunit ridge or the inner ridge of the beta subunit, and the bottom side here of the anti-parallel beta plated sheet are where the variability exists. So again, MHC class 2, it's kind of important that this works in this particular way because it needs the breakdown of this endocytic vesicle through the production of the phagolysosome. The phagolysosome can both acidify the endosome and introduce new peptides, peptidases and proteases that helps break down these down into amino acid chains and hopefully into small peptides. The clip protein that remains from the invariant chain has to be removed by HLA-DM. HLA-DM allows for the alpha subunits, for the beta subunits to separate. And then when the right peptide comes in, it recollapses those subunits, and then it is trafficked as a peptide-bearing MHC molecule to the cell surface. Whereas for class 1, again, these are for intracellular antigens. So we're talking about really viral antigen, normal human antigen, and cancer antigens. And what we see is the breakdown of these antigens via the immunoproteasome allows for the production of peptides. These peptides are retrogradely trafficked to the endoplasmic reticulum, where they can be loaded through the binding of calcium to calreticulin. Calreticulin opens, allowing for the peptide to shuttle into the MHC class 1, and as long as the peptide MHC class 1 variability region can coalesce, it is then as a fully composed molecule sent to the Golgi body, where it is then trafficked to the cell surface. So MHC expressing dendritic cells, as we talked about before, they reside in the tissue with other monocytes and such as like macrophages. When they are expressing MHC class 1 or MHC class 2, they're going to produce certain cytokines, including pro-inflammatory cytokines like interferons and canonical first cytokines like IL-12 and IL-6. And they're going to be doing so and expressing these molecules as they travel via the afferent lymph, the lymph node. Then they look for naive T cells again. If this is MHC class 2, they're looking for T helper cells, and these are your CD4. And if it's MHC class 1, this is for CD8 positive T cells. So PRRs, again, the vast majority of them, when they are engaged, are really important for the function of phagocytosis. The one lone difference being the toll-like receptors, which, again, there are 10 different toll-like receptors, but their job is not to engage a phagocyte or another type of cell with the ability for phagocytosis. It is actually non-phagocytic at all. When these particular pattern recognition receptors or the toll-like receptor pattern recognition receptors are activated, they change the signaling profile of the cell that has been engaged. So for macrophages that are activated, especially when they turn into M1s, they start secreting cytokines as they're killing bacteria or viruses or whatever. And the first 3 cytokines that they secrete are IL-1, IL-6, and TNF-alpha. These proteins work in a triumvirate to do a lot of different things, but the biggest things that they do are the following. They move and secrete these into the bloodstream so that they can get to the liver, which increases the expression of acute phase proteins, mannose-binding lectin for complement. So C-reactive protein is, these are pentraxins, these are mannose-binding lectin for complement, and in the liver they also increase the expression of actual complement and defense and expression. In the bone marrow, IL-6 plays a really important role in mobilization of neutrophils, but also played an important role in the activation of neutrophils. So mobilization and activation is really kind of a context, and we'll see this later, between IL-6 and CXCL8 or IL-8. IL-8 and CXCL8 all kind of combine to activate and home neutrophils. IL-1 and TNF alpha play an important role in regulating hypothalamic control of the calorimetry, functionality, and metabolism of your fat and muscle cells. So the hypothalamus can secrete hormones that is really important for increasing body temperature, but the increase in body temperature comes from the ability for an increase in energy mobilization in the fat muscles to produce energy, and then fast twitch, type 1 muscle function in order to generate higher body temperature, also known as a fever. So here's kind of a few of these things. So bacteria induce MAX to produce IL-6. Again, this is going to play an important role in both neutrophils, so neutrophils, and IL-6 travels to the liver to increase mannose-binding lectin, C-reactive protein, and again, these will help with complement. These will also help with the ability to spot or identify pathogens, so enhancing innate immune function. So C-reactive protein binds to phosphocholine, which is a phospholipid that's on bacterial surfaces, and it basically serves as a complement activator. And obviously, mannose-binding lectin also serves as a potent activator of complement as well. So here you go. C-reactive protein and mannose-binding lectin, these are really important for pathogen recognition. But again, the liver also produces complement components, which again, really important for pathogen elimination, including factor B, which is really important for the alternative pathway for the activation of complement. And then you have the expression of other important proteins such as GCSF, amyloid A, and phospholipases. These are all really important for activating the immune response and also activation of procoagulation cascades to help get the body ready just in case the infection travels to the bloodstream and causes bleeding or damage to the blood itself. There's a way to coagulate the blood in a very rapid manner. So, as we can see, when we're looking at the inflammation over days, within the week, we have really high peak, but then a very quick crash out of things like serum amyloid A and C-reactive protein. But then like as we progress the infection, or maybe post-infection, 14, 21 days out, we have an increase in expression of fibrinogen, which is important for clotting. So this would be really important for tissue repair. And then you see, things like complement receptor, there's a subtle increase, but then a decrease over time. And again, this is because complement can is always on. So you already have a good collection of complement proteins at your disposal, but it's also inducible. So you get this mild increase to increase the likelihood of complement functioning. And again, things like C3, uh, human cells are resistant to the formation of the membrane attack complex, but they're not immune to the effects of binding of C3 and C3 conversions of other parts of complement. All right, that does it for phagocytes. Thanks for watching.